Good afternoon, everyone. My name is Chong.
Hi, good afternoon. My name is Vinod.
Hello, my name is Ani.
Good afternoon. My name is Rizwan.
Assalamualaikum warahmatullahi wabarakatuh and a very good day. I greet to all the viewers. Today we are going to present a presentations regarding an experiment entitled Activated Carbon Adsorptions in relation to kinetics, isotherms, and continuous flow operations. But first and foremost, I would like to ask you, what is an adsorptions? Adsorption process is a process where active materials in true solutions are being removed from the solvent by interface transfer to the surface of adsorbent particles. There are several adsorbent particles that exist, but the common one used in industrial field is activated carbon. Activated carbon comes into several either fine powder or granular form of adsorbent particles, where they will have high concentrations of contaminants when reaching breakthrough points. There are basically three facts on steps of activated carbon. First, the substances adsorb to the exterior of the carbon granules. Secondly, substances move into the carbon pores. And thirdly, substances adsorb to the interior walls of the carbon. Reaching to the aim of the experiment. The objective of the experiment is to investigate the absorption properties of activated carbon in relation to equilibrium isotherms, adsorption kinetics, and operational characteristics of lab-scale packed-bag reactors. In this experiment, there are two parts, part A and part B. So with that, I will pass the floor to my friend Vinod to explain more. Thank you.
Such that the material needed is distilled water, methylene blue, and granular activated carbon. Next, I'll be presenting you about the experiment design. Well, each of the absorbent experiment here and after describe and employ synthetic aqueous solution of methylene blue as the test solution, and any other surface material or absorbent such as organic dye, phenol, or alkyl benzene can be substituted as the absorbent. So the natural waters and wastewaters can also be used as dissolved organics as a test solution, but provided that they have removed the particulates or any other content organism in these test solutions. However, if methylene blue is replaced with another absorbent, then it will be necessary to define an appropriate analytical technique for whatever absorbent it utilized. So now I'll move on to the procedure of this experiment. So this first step, you've got to prepare 1 liter methylene blue in 3 different beakers, and their concentrations are 10 mg per liter, 30 mg per liter, and 60 mg per liter. And after your preparation, you got to place that in the laboratory stirrer and start stirring it. So as you start stirring it, at time 0, you got to add granular activated carbon in those 3 beakers. In beaker 1, you got to add 100 mg. Beaker 2, you got to add 250 mg. And beaker 3, you have to have 350 mg. As in the first step, take samples in 10-minute interval and continue sampling until it attains equilibrium. If the granular activated carbon does not dissolve, then it needs to be filtered before sampling. And the last step would be determine the remaining methylene blue concentration by utilizing the calibration curve prepared before. And next would be methodology for part B, and that will be presented by Mr. Swai.
For the methodology part B, firstly, we set 11 set of 250 ml flask which containing 100 ml of 125 mg per liter of methylene blue solution. And the second one, all the flasks have been added with granular activated carbon with specific weight that have been assigned. And all these flasks have been covered with the parafilm, and it continue to be agitated, which means it will be stirred by using the laboratory shaker. And during this agitator, we are using about approximate 10-minute interval for each measurement, which means that after we stir it, then the remaining methylene blue concentration will measured for each flask. And lastly, the quantity of methylene blue have been calculated that have been transferred to the surface of activated carbon for each flask. And the step is repeating from this one, which mean with 10-minute interval until it give a equilibrium data.
Now I'm presenting on the part result and discussions. For part A, absorption kinetics, we can see here this graph is plotted based on the normalized solution phase concentration C over C naught Vs time. And this is the effect of the contact time on the absorption of the methylene blue at different time and different concentrations. Based on the graph, we can say that the extent of the color removal from the methylene blue by the activated carbon increased with the increase of contact time until the activated carbon becomes saturated. This is the point where the activated carbon becomes saturated. This is the 10 mg per liter of activated carbons, and this is the breakpoint of the absorbance, which is 1.739. For 60 mg per liter, the breakpoint is 2.617, and for the 30 mg per liter, the breakpoint is still not appearing after 60 minutes. We can see here there's a fluctuations in the graph for 60 and 30 mg per liter. This is because the error during the experiment at time 15 minutes and 60 minutes, such as the human error and the random error, for example, environmental, that caused the. And for part B, which is the absorption isotope, we are going to analyze the results based on two methods. The first method is the Friedrich equations. The graph is plotted based on the log Q vs log C. The Q is the amount of adsorbed per unit weight of the absorbance, whereas C is the concentration of the fluid, and K and N is the coefficient. The second method is the Langmuir equations. The graph is plotted based on 1 over Q vs 1 over C, where the slope is the K over Q naught and the intercept is 1 over Q naught. For the first graph in part B, a graph of log Q against log C is plotted as shown in the first graph shown. So from the graph we can say that the reading is almost constant at every log of the concentrations. For data to fit the equation, the Friedrich isotherm, the graph must be linear. However, in this experiment, the linearity is very small, and so we conclude that the result does not follow the Friedrich isotherm. And for the second graph, C over Q is plotted against the concentrations. And from the graph, we can see that the C over Q increase with the increasing of concentrations. And the plot of the CQ against C, as shown in the second graph, give a straight line for all the concentrations, and that implies that the absorption for the absorbance well fit to the Langmuir isotherm. And lastly, we conclude that the result do not follow Friedrich isotherm well compared to the Langmuir isotherm. Thank you.
I will be presenting about the industrial application of the activated carbon. First of all, activated carbon is widely used in the water treatment plant. In this plant, activated carbon is used in the purification of water for human consumption. Next, in the medical industry, activated carbon is used for the wound dressing, odor control, mask and filters. And lastly, in food and beverage industry, activated carbon is used in processing stage of the production of the food, including sweeteners, edible oil and other food products. And with this, I would like to conclude our presentation. Conclusion, adsorption is a unit operation that is very important to our life. The knowledge of understanding the use of activated carbon is very important in our engineering field. With that, I would like to end our presentation, and we would like to thank Dr. Taslima and Dr. Lukman for this wonderful opportunity. Thank you.